Actually, I'm not going to go into photosynthesis just yet. Change my start. I will start off, I will close off the chapter, the previous chapter first before we go into the next one. Okay, so you can stand by your notes on cellular respiration first. You download that? Yeah, one more stick. No, I'm the kind of person, right? Yeah. Well, if I want tasks, right, I'll break it down in many tasks, then I'll put many checkboxes in my book. This small thing I do or something. Makes me feel good. Okay, ready? I went how many? Press it, okay. Alright, today we're going to close off where we last left off in the previous lesson. In the previous lesson, we did a little bit of role playing, right? Where all of you perform a skit. I must say, you had quite a good grasp of what we termed as oxidative phosphorylation. That's the last step in cellular respiration. There's quite a lot of things that were happening. So let's watch a video to summarize the previous lesson. Alright, as we watch the video, again, the key things to think of when we look at interactions. Interactions, you want to know the where, the who, the what change. Okay, so we pay attention to this again as we watch through the video and we tease out all these points. Ready?
Most of the energy harvested from organic molecules during glycolysis and the Krebs cycle is stored in NADH. NADH and FADH2 molecules give up their high-energy electrons in the third phase of cellular respiration, electron transport and ATP synthase, where most of the cell's ATP fuel is produced. The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondrion. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of electrons to an oxygen atom and two hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed up hydrogen ions give up their energy when they diffuse through a special protein complex in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP. Produced.
Okay, so that was a quick summary of the last stage. You have this diagram in your book. Something similar. Yeah, I quite like this as a summary. It actually tells you what is needed, what is produced. If you look at the very top of it here, electrons carried by NADH. And the arrow shoots up, goes to the last step. If you put electron transport or oxidative phosphorylation, you have that arrow also moving upwards to where it's supposed to be. Yeah. Okay. So what are the byproducts of oxidative phosphorylation? At the end of all this, what will we produce? Okay, we produce a lot of ATP. Anything else? Okay, water. That's part of the product. Anything else that's produced? CO2. Okay, was CO2 produced at any step? Okay, not shown here. Actually, it is not. Okay, CO2 is actually not produced at the last step. CO2 is produced in the Krebs cycle as well as in the link reaction. So the CO2 we breathe out, right, actually doesn't get produced at this last step. What else is produced? Okay, FAD plus, right? No, actually it's just FAD, yeah? It's FAD and NAD plus, right? Where did these come from? It came it arose after it gave up its electrons to the chain, right? Okay. So in a sense, these fellas have been regenerated. That's great, right? You just recycle them over and over again. Because at the end of the day, when we look at this particular mind map over here, glycolysis, pyruvate oxidation or link reaction, Krebs cycle, they all require one thing to continue functioning. And that one thing would be NAD plus or FAD. Right? We need electron carriers at the end of the day. These electron carriers, otherwise they draw out the electrons at every step of the way. So it is very important that oxidative phosphorylation continues to its end, so that we regenerate these electron carriers to be reused at every step of the way. So if you really want to complete this mind map, what you can do is have one more arrow coming out and going back into Krebs cycle, going back into the second step, going back to the first step, and write NAD plus. Okay? That's where everything will feed back into. So you know which part of your notes has this particular image? Okay, while you're doing that, don't mind me, I need to find one slide I want on the screen. Nice. Okay, so now we've reached a problem. What if I put an environment where you have no oxygen? Right? We talked about this last week. Right? Oxygen acts as the final electron acceptor. All electrons that pass down each chain, the chain will end up finding an oxygen. But if you stop breathing, you remove this altogether, this chain is going to stop functioning, right? And if this chain stops functioning, you know what's going to happen? The NADH at the start has nowhere else to give its electrons to. Then we have a problem here. We can't regenerate this anymore. Right? And if we cannot regenerate NAD+, it's going to be a problem. That means every step that comes before now ceases to function too. Right? NAD+ is no longer being supplied to every step. So we have a problem here. That's why we need oxygen so badly. Okay? But actually our body has a way of getting around this. I'll let you try something right now. Raise your hand. you done? Oh, you watch the video already, yeah? Okay, so you haven't watched yet. Okay, so you haven't watched, right? You try this yourself. How long do you do it for? 1 minute? Okay. We do again, you raise your hand. Okay, now you just keep clenching, releasing. After a while, I'll see your hand start to slow down. Still going strong? I wonder if your CCA will affect what kind of, how long it can last. Ivan, right, it's like losing steam already. Do you feel the pain yet? Do you feel your arm cramping up? Oh yeah. Oh, still can go, not bad. Go faster. Come on. Yeah, I tell you, I think no more pass without context, right? I want someone to walk past right now. If someone walk past, right, we will collectively look to them. Hey, okay. Hey, you all slow down first. It'll be hilarious. Okay. So, you feel the cramp in your arm? Feeling it? Okay. So in a while, right, we're just gonna stump 3 girls that are gonna walk past. Okay, so now yeah. Okay, you feel the cramp in your arm? Yeah? You know what's building up in your arm right now? Yeah. You felt this pain before when you were made to run up a flight of stairs when you don't want to. Yeah? Lactic acid. Lactic acid is building up in your arms. Now, under normal conditions, actually, I cannot think of a scenario where your body will run out of oxygen unless I put my arm over you, my hand over you, right? And you not breathe, right? That's the sure way, definite way of you not getting oxygen. Okay? There's one other scenario where you will run out of oxygen, or rather, not enough oxygen for the last step to carry out. It happens when you are using up oxygen fast. And for example, if you go for a sprint, right, versus you go for a jog, chances are when you go for a sprint, you'll get cramps all over, right? Yeah? That's because when you go for a sprint, you're actually not breathing fast enough to supply enough oxygen to all parts of your body. This one is actually one scenario. When you are going for sprints or doing rigorous exercises, your body is actually not getting as much oxygen as it should. So actually, you are inducing a scenario where you are short of oxygen. But our body is able to make up for this. What does our body do? We call this fermentation. This kind of it. Our body can carry out fermentation. Okay, so where am I at? If you go to your notes, you scroll all the way down to the very last part. I'm at this part over here, fermentation. Okay. Our body can carry out something called lactate fermentation. Top left-hand corner, the word lactate fermentation. The beauty of lactate fermentation is this. It makes use of glycolysis step. Now we know that glycolysis produces net of 2 ATP, right? Okay, so actually our body is quite smart. In the event that we don't have enough oxygen, this is what it does. Okay, so we don't have any more NAD+. We cannot continue producing ATP, right? So our body does this instead. It makes use of the pyruvate that it produces during glycolysis. Then it converts this pyruvate into lactic acid. But in converting it into lactic acid, it would use up any NADH that was produced, convert it back to NAD+. Okay? So in a sense, at the glycolysis step, it can regenerate NAD+. And it just goes into a loop. You can let NAD+ take out more electrons, then you will use NADH to convert pyruvate into lactate, and then the NADH will form back NAD+, and it just is an endless loop. So, lactate fermentation is our body's way of coping when there's a lack of oxygen. So they can still supply you with a little bit of energy, 2 ATP, a little bit of energy for you to carry out then a little bit more of activity. But of course, we have a threshold. Your arm starts to cramp up after a while, right? That's because as lactic acid builds up in your body, it starts to cramp up all the muscles to prevent you from continuing the arm that you're doing. Because actually, when you lack oxygen, it's just not very good for you to continue on. Yeah? So we have a threshold. We do stop ourselves in a sense. That's inside the human body. Fungi and bacteria can also carry out this process. But rather than converting pyruvate to lactic acid, it converts it into something that we can eat and drink. It converts it into alcohol. You know how wine is made? Yeah? So actually how wine is made is this. They take yeast, they put it into grape juice. They know what they do, they run back to the bacteria, to the yeast. They just cover it up and prevent any oxygen from getting in. So they starve the yeast of oxygen. But then something magical happens. When it's starved of oxygen, it starts converting the pyruvate first into this chemical and then converts it into ethanol. But it will make use of the NADH and convert it back into NAD+. And by doing so, it supplies a little bit of ATP to let the yeast continue growing and multiplying. But that's how we get our alcohol, right? That's how the grape juice gets filled with alcohol, ethanol. Along the way, this chemical reaction also produces CO2. And that's how a lot of alcoholic drinks are also fizzy. Lots of bubbles inside. I've left grapes inside my fridge until this happened. Until each grape, right, I put in my mouth, right, it tells alcohol and then it's fizzy inside. So, I mean, you can try this, but when it goes moldy on the outside, you're going to stop. Mine didn't go moldy, that's why I just popped it in my mouth. It's an amazing feeling. Okay? And it makes me wonder, maybe that's why last time people, when they make wine, right, they will take grape juice and grape, right, and they just step into it. There's a lot of yeast in our feet, Yeah. So perhaps that's why they were stepping in a grape. I always wonder what the first person was doing when they created things like wine. Did they keep just a lot of grape juice? Very kiasu, keep a lot of grape juice. Then after it started to spoil, then they're like, okay, heck, I don't want to waste it, and then just drink it. Okay, so this last step, this lesson, the first part of this lesson is just showing you about anaerobic respiration. Anaerobic means if there's no O2, okay? If there's no oxygen, actually our body can still cope for a short while. How it copes is to hijack glycolysis and regenerate NAD+ by converting pyruvate to lactic acid. Okay? And bacteria and yeast also can carry out anaerobic respiration. Okay, give you some time to write down anything you want to write down in your notes. Or if you want to ask me any questions, please do so. Oh, wow. And what do you learn exactly about fermentation? Is it also the same thing? So your teacher is Mr. Low. Mr. Low did mention alcohol formation and stuff. Oh, okay. Yeah, usually when temperature is involved in biology, it has to do with enzymes. That's just the ideal optimum temperature. Yes, so some examples of instances where we make use of these forms of fermentation. So, for example, kimchi. Yeah, kimchi is a little bit sour, right? Okay, the sourness comes from lactic acid. So we leave kimchi out, and then deprive it of oxygen. Bacteria goes in, starts to carry out this form of fermentation. You get this sourness, tartness to your kimchi. One example is wine. Do you know what kombucha is? Yeah, also, no, kombucha I should put over here. It's a bit sour. Okay, so chances are fermented things that are sour likely went through this form of fermentation. And those with alcohol likely went through this one, like maybe beer, wine. Wow, okay, well let me list out the alcohol. Soju. What? Sake, wow, sake, gin. And guess what? I'm allergic to alcohol. So when I drink alcohol, right, like my airways start to close up, then I start to cannot breathe. So y'all don't try to spike me, can kill me one. How I know that? Okay, so one day, okay, so little amounts won't kill me but when I drink more and more and more, I realize, right, I start to wheeze. Like every breath I take in and out, right, you can hear a tight whistle, like yeah, it sounds like that. So I realized that I was allergic to alcohol. That's quite a good excuse. Every time people want to ask me like, hey, you want to go to the pub and like, oh, last time in like army or JC, right, after school, some of them want to like they want to go to the bar. You're not bar, the club. Then I'm like, I cannot, like I may die. And like, oh, okay. Then they tell me, oh, just go there and dance on the dance floor without drinking. Like, it's a bit weird. Might as well go ballroom. So I'm not dissing ballroom dancing. Okay, so we have closed off the chapter on cellular respiration. The next chapter on photosynthesis actually is largely almost similar. Okay? Because if you think about it, photosynthesis is all about producing glucose, right? And oxygen. So actually, let's go backwards. Photosynthesis is going, taking the process backwards. Yeah? Okay. Today, we're just going to start with photosynthesis. If I may get you to go into SLS first. Okay, today will be a light lesson. I'll be doing a demonstration right in front of you, right? Oh, shucks. I always can reset students' password. Who resets my password? Okay, so I'd like you to go to photosynthesis, which have been assigned to you already. Let's start off first with folder A. I just want to know what you know. Okay, we still adopt the same language of interactions. Where, who, what changed. And so in the case of photosynthesis, what do you know at the moment? What is needed in photosynthesis? I'd like you to key down, type down some of the things you know. Related to where? Where does photosynthesis take place? Who do you think is involved? And what will change? Okay, and then we'll just have a look through, a quick sharing. Once you type your response in, you may have a look at your friends' responses. child says light, carbon dioxide, water, and a plant. Rightfully so. Yeah, that's the way it takes place. Okay, so light is also needed. Water, okay, indeed, water is needed. At the same time, we need light. Ian says we need light, and he specifically gave a little bit more UV light. Just to share, actually plants make use of a lot of spectrums of light, other than one color. I want to make a guess. Yeah, green. Plants aren't able to absorb green light. That's why they are green. Yeah? Okay, but then notice some plants are purple. Yeah? So that means they just cannot absorb that form of light. So can you imagine in a world or alien world where a plant cannot absorb another color, you can expect that plant to turn out a wildly different color. And if we go back to the evolution of plants, there's actually bacteria. Bacteria were the first organisms that were able to photosynthesize. And you know the chlorophyll they have is purple in color. So these bacteria appear purple. Plants arose after these bacteria. So in order not to compete with these bacteria, right, they decided to absorb light from other spectrums. So what's left is what we see being reflected into our eyes, the green. The tree is very green. A lot of algae has grown onto it. Okay? Other than that, light, chlorophyll and chloroplasts, right, Debbie? Okay, if we say that light is needed, therefore we need to be able to absorb the light in some way. Right? And where do we absorb the light? We absorb it in our chloroplasts. That's something we're going to look at today. In the structure of chloroplasts. I think we've seen the structure of mitochondria quite often, but structure of chloroplasts is quite an interesting structure. And inside our chloroplasts, we have chlorophyll. So what's the difference? Here we're going to find out in a while. Okay, but long story short, is the pigment found inside chloroplasts. Chloroplast is an organelle that stores chlorophyll. Okay, Manas says sunlight, chlorophyll, water, carbon dioxide. And largely the same as we scroll down. Let's see if anything different as scroll up. Let me refresh. A lot more now. Since Xiao says to perform photosynthesis, plant needs three things: carbon dioxide, water, and sunlight for photosynthesis. Even bolder it for us. Okay, what are the products? Okay, let's look at Natalie's response. Where it takes place in the plant. Actually, very specifically, it takes place in the chloroplasts. What changes? Okay, she's taking the lens of interactions. Chlorophyll converts CO2 into oxygen. Not exactly, it doesn't convert it into oxygen, it converts it to sugar. Oxygen is a byproduct of it. Okay? This is the general equation for photosynthesis. If you look at your notes, this is what you'll see also. Okay, so largely I find that we have hit the key ideas of what is involved. You all learn this in primary school? Yeah. Okay. Actually in primary school they all learn structure of flower, this kind of thing. Yeah, so you know what stigma is. All of it. Like good or bad? Sorry, why bad? Usually, when it comes to plant reproduction, you all will learn plant reproduction again this year. When you come back in term 2, you'll learn plant reproduction. That's human reproduction. Okay, now I would like to perform a demonstration in front of you. Okay, well, before I perform the demonstration, you can stand by your notes also, so that you can notate if you want to. Sorry, not if you want to, you should. begin today, could I just get you to read this page, an introduction to photosynthesis. Just get a broad idea first. Get an introduction to photosynthesis. Then we'll go into the next part together, chloroplasts. Done reading, maybe look up. Continue reading. I just need you to read the overview. We begin our topic of photosynthesis by looking at the structure of chloroplasts, just like what we did for mitochondria. Yeah? The chloroplast is quite similar in that it has outer membrane, okay? Outer membrane. It has an inner membrane. But what makes chloroplasts different from mitochondria? Number one, you notice the inner membrane is not highly folded. Okay? Number two, it actually has a third layer of membranes, right, that folds upon itself to form many small little like M&Ms. So this is what you see in a textbook. Lots of small little structures that look like that, like Mentos stacked on each other. Each one of them we call a thylakoid. Okay, so the vocab for this chapter, thylakoid. The membrane of the thylakoid is called the thylakoid membrane. Okay, second vocab. last two vocab. The inside space of the thylakoid is called the lumen. Lumen of thylakoid. Okay, so three vocab there. Thylakoid are all these small little M&Ms. They are enveloped by it's made up of this membrane called thylakoid membrane. The inside space is called the lumen. Okay, one last vocab. This space surrounding the thylakoids is not cytoplasm. It's what we call stroma. Okay, so with these additional four, helps you describe the chloroplast a little bit better. Okay. Why is it important to know this vocab? So that when we go through the various parts of the chapter, we know where. We know where everything is taking place. So if I ever say the stroma, mentally you should visualize the areas outside. When I say the lumen of thylakoid, mentally you have to visualize the inside spaces. Okay, that's the value of knowing the vocabulary. No, it's called grana. G-R-A-N-A. Grana. Fluoro is granum. Wow, I don't know that. Is it saying that? Granum, grana? Granum. Grana. Fluoro is granum. You help me Google? Difference between grana and granum. So fluoro is granum, is it? So granum is many steps. Grana is one. So you can see, I very rarely make use of the word grana in my description of photosynthesis. Because we zoom in mainly to each individual M&M. Okay, so that's just one part for today. The structure of the chloroplast. Let me draw another box so I can fix it. Okay, structure of chloroplast. That's the first step for today. Next, let's try to understand a little bit about how chlorophyll pigments work. This is a chlorophyll pigment. Notice it's just a very complicated chemical. Okay. To understand how chlorophyll pigments work, we need to understand a little bit about electrons. Okay. If you go to the next page of your notes, you will see you'll put this circle over here, chlorophyll molecule, and then they'll show an arrow shooting up representing electrons and then going back down. Okay, I'd like to perform a real demonstration right now. Here I have high energy light. My physics is not very good, but I remember my physics teacher teaching me UV light is quite high energy. Yeah. Okay, so this UV light is quite dangerous also. Every year when I perform this demonstration, I accidentally shine a bit on my arm, right? Then I go home with itchy. Okay, so this UV light is quite powerful. It can cause cancer, skin cancer. Okay, it messes up cell division. Okay, so if I were to switch on the light, actually cannot quite see much. Okay, because it's very high power, you only see its effect when it shines onto things. Okay, on top of that, I have this thing called tonic water. Anyone drinks this? Have I drink before? Do you like it? It's just normal water, but? Okay, it's like gassy normal water, right? Okay. But if you look into the ingredients of tonic water, I noticed that it says it contains quinine. Okay? It just puts right here, you know, underneath, contains quinine. Now, quinine has a very special property. Much like chlorophyll, its electrons are excitable. What do I mean by excitable? Okay, let's say I have electrons inside the chloroplast of quinine. If I were to shine light, high energy light onto these electrons, something amazing happens. The electrons get excited. Okay? It gets excited, filled with energy. At the same time, after it gets excited, it will relax. It will go back down to its non-excited state. It is like a cycle. When I shine light, it gets excited, and at the same time, it goes back down its non-excited state. It's like it's jumping. Okay? But something amazing happens when it comes back down from its jump. Okay, I'd like to switch off the light right now. Can I get a volunteer to help me out with this demonstration? Okay, wait, let me ask someone that's not, I already called. Kylie. You help me out with my demo? Okay, class, welcome Kylie. Yeah, what am I doing this slide? Kylie will help me with, okay, so Kylie, all I need to do is this, open this can, okay, in a dramatic way. After that, I would like you, so I'm going to balance this glass vial onto my high power UV light. I'm going to switch on the UV light, okay? What I need you to do is to switch, open this, and then you aim it here. Don't pour over my hand, don't pour into the light. Cannot? You do that? Okay. So, Heine's going to pour in some of this quinine. The moment it lands into this glass vial, the UV light, the high powered UV light will strike the quinine and excite the electrons. Alright? Now let's watch what happens when the electrons get excited. Yes, that was dramatic. We pass. Yeah, probably more. Okay, thank you. Yeah? So, here is the phenomenon of fluorescence. Yeah? The idea that certain things can glow when supplied with high energy light. If I were to take this out of the UV light, it actually doesn't have much of a property. But the moment I bring in high powered light to the quinine, the electrons get excited. It's like it's jumping up and down, but every time it lands back down, it releases energy in the form of light. Without this high energy light, the electrons aren't jumping. But when I put it back here, it makes a high jump. Then when it lands back down, it releases light. This amazing thing about quinine. Okay, thank you. You want that drink? Anyone wants this drink? Keep it. Okay. Okay. Dongchen, you can have the drink. Yeah. What am I doing? Okay. So that's our first key idea for today. That electrons can be excited. They are excitable. But the second thing is that when they return to ground state means it's unexcited state, it will release energy. Okay. When an excited electrons returns to ground state, it will release energy. This energy can take many forms. Could be thermal energy, but in this case, we observe light energy. In this particular instance, we saw that it was released as light energy when it goes back down to ground state. Now, earlier on I said that quinine has the same properties as chlorophyll. So I've extracted out chlorophyll. Yeah? Here I have a vial of chlorophyll. You see it's green when I swirl it around. So I have a lot of chlorophyll here. It's like quinine. So if I were to shine high power light onto it, I should be able to excite electrons. I want to make a prediction what color it will fluoresce. Yes! Okay, it's right there on the screen. But I thought it would be quite magical to watch it in real life. Okay? The color you see won't be quite what you see in the image. I think that one is like saturated. But this is what you'll see in real life. Can you see? Can you see it? Looks like blood, right? Can you see? This one, it fluoresces a rather deep red, a bloody red effect, if you look at it. Can you see it or not? Looks at the back. Maybe you can see. Yeah? Okay. So this is what chlorophyll looks like when if I were to shine high power light. The electrons are being excited. It's also going back to ground state. But when going back to ground state, it releases light energy. And the light in particular is red in color in this case. Yes?
But if the same amount of light energy is provided to the electrons, why
if the same amount of electrons, oh, why would they go back down to ground state? If I constantly supply, is it? Oh, that's an interesting question. I don't know. I think that once there's a threshold of how much it gets excited. So I think once the electron is excited, it cannot gain any more, then it will return back down if it wants to gain some more. So, okay, so that's the key idea, that electrons can be excited. And the same thing happens in the chlorophyll pigments. But have you ever wondered, right, if I were to shine this onto our leaves, will we see glow the same way? Yeah? Okay, so let's plug the leaf up. Yeah, so I have a brass here. You just see it happens the same way. Anything? No? Why it's not turning red, right? What this means to say that the energy that's being released from the excited electrons is going somewhere. Right? This leaf right here is not wasting the energy in the form of light being released as fluorescence. Instead, it is using the energy for a particular purpose. And that's actually what chlorophyll is doing. Chlorophyll pigments make use of the idea that electrons can be excited. But when it goes back down to its ground state, rather than let the energy get released in the environment, it uses it. It captures the energy for a meaningful purpose. And we'll see what purpose it serves in a while. What do with this? Don't show you what? Okay, so that's the first part. Then again, you take down notes if there's anything meaningful you want to take down. Bit more about chlorophyll. Let's talk about where we find chlorophyll. We need to now understand how the leaves are able to harness the energy. Okay? If I were to look at the thylakoid membrane, okay, that's the new vocab for today. The thylakoid membrane. Actually, dotted along the thylakoid membrane are these funny looking structures. These structures look kind of like an orange that has the skin peeled off and has all these, what you call each segment of orange. Okay, it looks like a peeled orange. Lots of segments surrounding a core. Okay, we call each one of these segments light harvesting complex. Okay, each one of the segment is a light harvesting complex. So that means there are many light harvesting complexes here. Each segment is one of them. I said also that there's a core that all these light harvesting complex surrounds. We call that the reaction center complex. So reaction center complex. You see the word complex, what does it mean? It's not that it's very complicated. It means to say that it's a mixture of many proteins. Okay? So this light harvesting complex made up of many proteins, same as the reaction center complex. There's only one reaction center complex, but there are many light harvesting complex surrounding it. So where do we find the chlorophyll? Let me dissect this entire structure apart. Okay, maybe one last vocab. Collectively, all of them together, reaction plus light harvesting complex, we call it a photosystem. But now let's break apart this photosystem and look on the inside. If I were to dissect this photosystem in half, like chop it, then look inside the photosystem, this is what you see. You see all the blue magnets I'm using here? The blue magnets represent chlorophyll pigments. So you find all the chlorophyll pigments like the juice or the orange, the segments, they are inside the segments. Okay? You can find a nicer image on the next page. This is a photosystem that's been sliced in half. You see all the green chlorophyll there? Yeah, so you find all the chlorophyll inside each light harvesting complex. A series of them. So how does this photosystem make use of the energy that's coming in from the light? It makes use of it in a very interesting way. You need to follow me for this very last segment. Some students get confused at how the energy is used, but I try my best to make it as animated as possible. First and foremost, I need to draw your attention. When you see this black light, you can go and highlight this transfer of energy. Okay, some students think it's electrons being transferred. It's not. It's energy being transferred. But how is energy being transferred? Okay, so this is how. So imagine now, light. Here we are. Wait for one more. Make a drastic attention. Okay, so this is how the light harvesting complex collects energy. So imagine now I shine light. Oh wow. Okay, so there are chemicals inside our paper that can fluoresce also. So I shine light onto the photosystem. So this is what will happen. The electron will get excited, right? So now to represent the excitation, I'm going to make it jump. Okay, but towards us. Okay, so like, woo. Okay, so that's excitation. Okay, so it's excited. Yay! Okay, but then it needs to come back down, right? So it's now going back to ground state. Okay, now it's not excited anymore. But where did the energy go? Okay, so the energy is used to excite the electron, the chlorophyll next to it. So I'll show you again. It gets excited. Woo, and goes back down. But as it's going back down, transfers the energy to the next electron to get excited. Yeah. Then it transfers it to the next one. Woo. Okay, here they are. So that's what's happening at the light harvesting complexes. You just need to, the first chlorophyll at the very top, the electrons just need to be excited. Every time it makes that jump back down to ground state, it passes the energy on to the next one. Kind of like, you all have ever done a Kallang wave before? You know, like, right now, right, if I were to if I'm the electron and then I do this, right, then I come back down, right, then in front of me, right, you do the same thing. Whoa, okay, the chlorophyll pass energy kind of in this way. We transfer the energy, not the electron. Okay? Okay, so that's the idea here. So it's the bottommost chlorophyll. Okay, then you look at this special pair of chlorophyll A molecules. Now imagine this. There are so many light harvesting complex surrounding the reaction center, right? So what it means to say is this. There are many chlorophyll that is collecting energy, passing it down to this one single pair of electrons right here. I know what's going to happen right here. Imagine if every segment channels all the energy to this electron over here. This electron will have too much energy, to the point that it doesn't just jump up, it will make a leap of faith it actually jumps out of its chlorophyll altogether. It goes out of its chlorophyll up to the very top. Okay, it's labeled there as a primary electron acceptor. Okay, so the key idea for how a photosystem works is this. Energy is transferred almost like a Kallang wave, but please don't write that down. And all the energy is concentrated into this electron here that gets excited to the point that it's transferred to this space on top called the primary electron acceptor. Still okay? And then the next question is, I pass to them for what? They pass to them for this reason. Remember our electron transport chain from last week in cellular respiration? Plants have exactly the same thing. So, from the primary electron acceptor, the electron will just get passed down the chain. From the primary electron acceptor, it will just pass down the electron transport chain and cause ATP to synthesize ATP synthase. Just like what we learned last week. Okay? So the only complicated part about photosynthesis is understanding how this particular thing works to release electrons for the chain itself. I will stop here today. Okay, we'll talk a little bit more about the chain next week. But what I need you to understand at the very least today is how each of these photosystems can release electrons. Okay? But the idea is that it releases electrons because it has gained so much energy from the sun. If you like to, stand by for next week's lesson. We only have one more lesson we do next week. You can read up ahead. Okay, so I will see you tomorrow. Wait, what? Yeah. I'll see you tomorrow, right? Okay, but tomorrow we're going to the lab for practical. Okay, so if I need to bring your goggles, we'll do a lab on photosynthesis. Okay. I will let you use the microscope again. You get to look at plants. At the same time, we will work with chlorophyll. Okay? So tomorrow, please bring your goggles for your lab lesson. Right, I want chlorophyll again in your eyeball.